Genetics Day 7. AIM. How can we analyze DNA? I know that to analyze means to look at something closely. So today we'll be talking about looking at DNA closely. Moving on into Do Now question 1. Number 1 says, experiments revealed the following information about a certain molecule. 1. It can be broken down into amino acids. So here, if something's being broken down into amino acids, I know that amino acids are the building block for proteins, right? 2, it can break down organic molecules into subunits, all right? So breaking down, so it seems like it's a reactor of some sort. And 3, its function is severely reduced if it is exposed to high or low pH conditions. So it only works in certain conditions. This molecule is most likely, it's not a hormone, not an antigen, not a nucleotide, definitely an enzyme. All right, 2. Define the molecule selected above. All right, you can take a second now to pause the video and complete number 2. For number 2, you should have had something similar to this. An enzyme is a biological catalyst which speeds up chemical reactions within the body. Part A, comparing DNA. For this, we're only going to think and share. We will not pair until we have our live stream. Question: why do you think scientists might want to be able to compare the sequences of DNA between two people? Pause the video now and jot down some reasons as to why you think comparing DNA sequences is important for scientists. A few reasons I have listed below are to establish paternity, learn about ancestry, and solving crime. There are many other reasons. I just limited my answers to 3 for the sake of the worksheet. Now, the key technique in establishing paternity and learning about ancestry is a process called gel electrophoresis. This technique is used to separate DNA fragments of different lengths. So essentially, your DNA fragments are being separated by size in order to show a relationship of some sort. This technique is able to move DNA fragments using an electrically charged gel. Now we're going to talk about the gel electrophoresis procedure. Step 1. DNA samples are cut into fragments using restriction enzyme. A restriction enzyme is an enzyme which recognizes and cut specific sequences in DNA. Okay, here we have another think and share. For this one, we're going to work with a restriction enzyme. This restriction enzyme that we'll be using cuts between bases G and C. All right? So with the DNA sequence that we have below, we're going to indicate where the enzyme would cut the DNA sequence and determine the number of DNA fragments. The restriction enzyme we'll be using only cuts between bases G and C. So in looking at our DNA sequence below, whenever I see G and C, I know that I'm going to make a cut. I'm going to start with reading my first DNA sequence. So I have A, T, 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 G, C. Okay, so I see a G and a C, so I know that that was terrible. I know that I'm going to make my first cut. Now I'm going to continue reading my DNA strand. C, T, A, C, C, A, G, A, C, G. So we have a CG, but because it's not GC, I am going to leave that alone. TTT, ATGC. So I see GC again. Now I've made all of the cuts. In order to determine the number of DNA fragments, I'm simply going to count in between each cut. So this would count as 1, this would count as 2, and this would count as 3. So in total, I have 3 DNA fragments. And again, I was able to find my DNA fragments by using my restriction enzyme and making cuts between the bases guanine and cytosine. Wherever I saw a G and a C, I knew to make my cut because this specific restriction enzyme made those cuts there. Okay, so if you look at the bottom or the middle of the worksheet, I created my own gel electrophoresis chart. Now once the restriction enzyme is put into the DNA and the DNA is broken up, the DNA is then pipetted into a special sponge-like gel. On my gel electrophoresis chart, if you look at the arrow or the laser, I'm circling around the wells. These wells are where the DNA is inserted. So using a pipette, a scientist will insert each sample of DNA. Once the DNA is inserted, electricity is then applied to the gel, making one side of the gel negative and the other side of the gel positive. So again, if you look at my diagram on the screen, I have indicated where the positive and negative parts are. The negative portion will be closest to the wells. Naturally, DNA has a negative charge. It is naturally pulled toward the positive side of the gel because opposites attract. The fragments that travel the farthest are the smallest. And as the DNA fragments are getting trapped, they form visible bands. Stop and jot. After digesting the DNA molecule with restriction enzyme in step 1, how many bands would you see if those fragments were ran through a gel electrophoresis test, and why? If you remember, with that restriction enzyme, we only had to cut in places where we saw bases G and C next to each other. In that sequence, we only had to make 2 cuts, resulting in 3 DNA fragments. Step 5. If DNA fragments from 2 samples settle in the same places, then the segments of DNA are common. The diagram on the left illustrates a gel electrophoresis test comparing DNA fragments from 3 men and a baby. Based upon the results of the test, which man is the baby's father? Explain how you know. Take this time now to pause the video and try to determine which man is the baby's father based on everything we just reviewed. You should have picked man 3 as being the baby's father because man 3 shares the most bands in common with the baby. If you look at the baby's results here and man 3's results here, you'll see that they each share a band here. They share this band together. They also share this band, these 2 bands, and this band here. So with them sharing those bands, that indicates a relationship between them. Part B, class practice. Base your answer to questions 1 and 2 on the information below and on your knowledge of biology. In an investigation, DNA samples from 4 organisms, A, B, C, and D, were cut into fragments. The number of bases in the resulting fragments for each sample is shown below. Number 1. The diagram below represents the gel-like material which the DNA fragments move during the gel electrophoresis. Draw lines to represent the positions of the fragments from each DNA sample when electrophoresis is completed. So what I'm going to do is, for sample A, I'm going to do that one with you guys. So if we're looking at sample A, we're looking at all of this here. Okay, and we're also working with this column here, or this row. So, the number of bases in DNA fragments, there are 4 different numbers here. We have 3, 9, 5, and 14. What I'm simply going to do in sample A is I'm going to find line 3 and make a line there, even though that was clearly on 4. Try again. 3. My next number is 9, so I'm going to place a line on 9. My next number is 5. I'm going to place a line on 5. And my last number is 14. And there we have the DNA fragments for sample A. For sample B, I'm going to do the same exact thing. Sample B starts with number 8. I'm going to go to 8, 4, 12, and 10. Now my lines aren't the straightest because this is like being done virtually, but you get the point. For sample C, I start with 11, so I go to 11 there, then down to 7, 6, and 8. And last but not least, for person D, we have 4, 12, 8, and 11. Number 2 is asking which 2 organisms are most closely related. Support your answer using data from this investigation. So, after comparing this gel electrophoresis test, I can clearly see that there is a relationship between sample B and sample D. They share the most DNA fragments in common. Part C, independent practice. Briefly summarize what you have learned in today's lesson about gel electrophoresis. You may wish to discuss what gel electrophoresis is, how it works, and what information scientists can gain from using it. Please have this information ready for tomorrow's Zoom call. I will make sure to ask everyone to give their own version of a summary at this part. So this will be done on the Zoom call. Okay? Base your answer to the question on the diagram below and on your knowledge of biology. The diagram shows the results of a technique used to analyze DNA. Number 1, the laboratory technique is known as gel electrophoresis. Gel electrophoresis. Number 2, this technique used to analyze DNA directly results in 2, separating DNA fragments based on their size. Number 3, the arrow represents the direction of movement of the DNA fragments. What is responsible for the movement of the DNA in this process? Electricity is responsible for the movement of DNA in this process. Electricity. Question 4. Identify the substance, organic compound, that was used to treat the DNA to produce the fragments that were put into the wells. So, they're talking about an organic compound that was used, so I know that it's definitely going to be a restriction enzyme. The restriction the restriction enzyme is responsible for the fragments that are created. Number 5, place a circle on the diagram above around the smallest band of DNA. So the smallest band of DNA would definitely be found near the bottom. Remember, the wells are up here, and the wells are near the negative charge. The bands that are towards the negative charge are the larger bands. The smaller bands are going to move the fastest, and they're going to be pulled toward the positive current. A group of scientists recently found members of a plant species they did not recognize. They wanted to determine if the unknown species was related to one or more of 4 known species, A, B, C, and D. The relationship between species can be determined most accurately by comparing the results of gel electrophoresis of the DNA from different species. The chart below represents the results of gel electrophoresis of the DNA from the unknown plant species and the 4 known species. Number 6. The unknown species is most closely related to which of the 4 known species? Support your answer. In order to answer this question, you have to analyze the placement of fragments within the gel electrophoresis test. So we're comparing these fragments here of the unknown species with the fragments of each species A, B, C, and D. Now, if we compare each band or each fragment of each species, we'll see that species C has the most relation because they have the most common bands. There are 1, 2, 3, 4 bands in common. So I would answer this question saying that the unknown species is most closely related to species C because they have 4 bands in common. A scientist was analyzing a DNA sample taken from a crime scene with DNA samples taken from 3 suspects. The scientist chose to compare the DNA samples using gel electrophoresis. She obtained the results below. Though puzzled at first, the scientist recognized that she had forgotten a key step in the procedure. Number 7. Based on the scientist's results, what step did she forget in her procedure? So there are 2 things that she could have forgotten to do. She could have forgotten to add the restriction enzyme, which is responsible for breaking up the fragments, or she could have forgot to power on the electricity. Without the electricity, there are no currents being sent, which will make the fragments move. So for number 8, using your answer to question 7, explain how failing to include the step you identified led to the results the scientists observed. So if the scientists didn't add in a restriction enzyme or electricity, there would be no movement from the DNA fragments.